basketball star from Osseo and UW Eau Claire alum who did research in chemistry here at UW Eau Claire with Dr. O. He's Director of Research and Innovation at Mayo Clinic Health System in northwestern Wisconsin, and that's been for about 6 months?
Yeah, 6-9 months.
Right now. And working to foster research collaborations between UW Eau Claire and Mayo staff and clinicians. And that's in addition to his ongoing research in regenerative medicine, and that's what he's going to talk about today. So I'll pass it over to him.
Thanks, Karen. Thanks for everybody coming. So I thought I would try to blend a couple of things together here because I think there's multiple topics of interest. I know, and my battery's 10%, so we have to talk fast. So that's, did that show up there? So I'm going to start out talking a bit about our research and be a little geeky and nerdy about some of the science and technology that we work on. But I also want to then transition that into how I see the collaboration between the university and Mayo can fill some of these gaps. And so I'm actually going to show a slide, a research slide at the end that is an unpublished slide that we're working on, just got it the other day, and I think it's an example of how we can start thinking about university faculty and students at UW Eau Claire interacting with some of these more complex health teams that we have at Mayo Clinic. And I think there's a gap there that is a challenge to bridge. And some of us that are willing to think idealistically or say, oh, that gap we can bridge. Those of us that are a little more pessimistic and reserved and whatever you want to call it, will say that gap, how do we bridge that? And so I think that it's an interesting debate, and I'm eager to learn from all of you as we continue to try to forge ways of bridging this gap between these two wonderful institutions called Mayo Clinic and UW Eau Claire. And I firmly have a high degree of confidence that we are going to do some remarkable things in the near future together. And I love the slogan of UW Eau Claire, the power of and. You probably all think it's kind of hokey at some point, and as we all do, we at Mayo Clinic say the needs of the patient come first, and it kind of becomes cliche and you kind of take it for granted. But I really, really love the power of and that's what this is about. It's the power of Mayo and UW Eau Claire, and how do we leverage it in ways that none of us have thought about. And for the last 6 to 9 months, it's been a lot of fun to learn and listen to what's going on here and to start brainstorming. And this is a continuation, I hope, today of that ongoing discussion. So to dive into the Old Hearts, New Problems, let me just give you a bit of a background of who we are. Has anybody ever seen me show this slide? So it's fair for me to ask what this picture is. Anybody willing to venture what this picture is in the middle? Lightning, that's a great one. Thanks for sharing. That's a pretty common one. It's even great example because you're flat wrong, so we can learn. So now that I set the stage so bluntly, anybody else willing to go for it?
Like a rocket.
Rocket's another one, and you're getting a little bit closer. Can anybody connect these dots? And why is there 2 rockets here? I'm teeing you up here. 2 rockets in the same picture, right? You see 2 lines, right? Your bifocals can see that, right? It's 2 lines there. So this is actually SpaceX. Anybody ever heard of SpaceX? SpaceX is anybody not heard of SpaceX? Be brave, all right. SpaceX is this flight exploration company that has been charged with trying to create a reusable rocket, if you will. So this is one of the first rockets that 3 years ago went into outer space. And you're looking at a 12-minute time-lapse photo here. So the rocket takes off, goes in and does its thing, and then comes back and lands in the same spot. That's pretty cool, right? So I always like to tell people, at least what I do isn't rocket science. It's a lot simpler than this. But think about this for a second. This isn't just a bottle rocket in the backyard. This rocket is 20 stories tall, 20 stories. It delivered 20 ton of cargo into outer space. After it delivered that cargo into outer space, it flipped itself around and flew itself back and landed 12 minutes later where it took off. I mean, think about that. It's remarkable. So how do you engineer something to do that? What did NASA never do that SpaceX did? Well, the weight to thrust ratio of this rocket had to be twice, because you had to lift twice the amount of rocket fuel to be able to fly 2 missions without refueling. So that weight to thrust ratio had to be re-engineered so you could still deliver cargo and not just fly fuel both ways. So the idea of this is the definition of research and innovation, and this is why I used both of these words in everything we talk about. Research is the process that converts money into new knowledge, and that's what universities are really good at. Research is the process that converts money, grant money, into new knowledge. Innovation is the process that converts knowledge into new money. Okay, that's innovation, that's the products, that's what healthcare does, honestly. We take care of sick patients, and we do it more and more efficiently and better and improve the outcomes, and we make money doing that. And when we make money, we reinvest that as a nonprofit into more education and research. But this ecosystem of research and innovation working together is really quite paradigm shifting for healthcare. If you think of Apple, if you think of Seagate, if you think of other industries, they do this all the time. They invest their money in research to create the knowledge that then they convert into new products. If you think of academics, we have a mission of new knowledge that we don't necessarily have to translate into a product, and there's an important component to that in a university structure. On a Mayo Clinic side, we dream about how to integrate this. How do we create the knowledge that we need to actually improve outcomes of patients? And I use this picture to say that the new generation, some of those sitting in this room, will be doing this, where the old generation never did this in healthcare. This is what NASA didn't do and what SpaceX is doing. And this is the paradigm for us to think in biomedical research of what we can do differently. Just to give you one more image here, what does NASA's flight control look like in the Apollo mission? Describe what you see. Apollo, 1950. Many of you in this room weren't even born. I wasn't either. What did 1950s look like when Apollo? What did you see?
White men and white shirts and ties.
White men, white shirts, black ties, right? And black
Pocket
frames. Pocket. You got that? Those young people in the room, can you even?
Slide rules.
You remember this picture? What did SpaceX mission control look like a few years back when this happened? What did you see there? What was it? Probably digital. It was actually, I've got a picture of this, and I love this. I need to put it in my slides. I say it and then I don't show it, but it's kids, hoodies, and these game station headphones with mics, right? That you'd think these kids are probably playing games in the basement of their parents' house, right? That's what mission control looked like doing that. Right? It's a new generation. It's a new way of thinking. It's a new people doing it. It's people thinking differently to make this happen, that what NASA was capable of doing. And that's not a verdict on NASA. NASA was doing things at a different time for a different purpose. It's just this is the reality of where industries are going. Where's the SpaceX in healthcare? Where's the mission control for the new healthcare? I think it's in Eau Claire. I think it's in collaborations between medical institutions and universities that are thinking differently. So this is the paradigm that is a bit grandiose, I will admit, but I think nonetheless is a vision that we should challenge our dogmas against. So the brief story of what we do is in hypoplastic left heart was really founded by Todd and Karen Wanek, the owners of Ashley Furniture, that really allowed us to start thinking how in healthcare, in a focused problem, can we tackle this? And so those of you that don't know, congenital heart disease, or specifically hypoplastic left heart, is a great place to pioneer this philosophy. This is the most severe congenital heart defect that we have in the United States. It requires 3 open heart surgeries, the first one at 3 days of life, the second one at 3 months of life, and the third one at 3 years of life. So think about that. For these kids to have a shot at life, they have to have 3 open heart surgeries that replumb their heart. That sounds remarkable, and it is remarkable because who was born before 1980? It's amazing to me some people don't raise their hand to that question.
It's hard to do the math.
Born before 1980. If you had this condition, you would not be sitting here today. Okay? Those of you that raised your hand, if you had this condition, you would not be here today. Those of you that didn't raise your hand are young enough that you could go through these open heart surgeries. This is your gift. This is your privilege. But now let me tell you the other side of the coin, that those of you born after 1980, those of you born today, have a 50% chance of seeing your 5th birthday. 50% chance of seeing your 5th birthday, and we think this is called healthcare. We think that this is innovation. Now that is innovation. This 3 surgeries are remarkable. But the fact that 50% of these kids don't see their 5th birthday is the reality. So who are we? We're a dedicated team of cross-functional teams that are really with the mission of delaying or preventing transplant for these kids. Because these kids are really the kids that are going to only have a chance in any measurable quality of life, long-term predictability that they'll get a transplant at some point. As a side note, how many heart transplants do we do in the United States? Throw a number out. How many heart transplants, all hospitals, all comers? Throw a number out. 100? You're so pessimistic, Karen. How would other people think? 8,000. What other people think? This price is right here. Don't go low. 100 to 8,000 is a big gap. Or is 8,000 too many? Does anybody think 8,000 heart transplants is a small number? The number is 2,000. 2,000 hearts get transplanted for all comers across the country under the age of 70 for any condition. 2,000. Just in the U.S. every year. That number has been the same for about a decade. It hasn't increased in a decade because of the obvious organ shortage. This condition, there's 1,000 of these kids being born every year. Okay? 1,000 of these kids. We only transplant 2,000 for all comers. You can start seeing why we exist. Here's some pictures of some of the reasons why we exist. How do we succeed? It's really redefining the problem. It's taking this old paradigm of surgical interventions that we think is a success, and those that do it today are very proud of it, but we redefine the problem. Is living to your 5th birthday 50% of the time success or failure? We think that's failure. That was success only a few years ago because that's all we had. So we're redefining the problem and not tolerating what we used to call a success. So our mission is pretty clear. It's solutions for HLHS, and how do we do this? Really encompasses about 60 people now within our close team and about 100 people across the country. So this is not a small team of people. And this map is really putting on it the idea that basic biorepository research, molecular mechanisms of disease on the left, with the idea how do we translate that all the way to transforming the care? How do we create new therapies? This is a grandiose vision that some would say is totally naive and some would say is a complete waste of money, but this is what we started out with 8 years ago. This is an 8-year-old slide. And we broke ourselves into teams of imaging, genetics, stem cells, and preclinical. And we recognized, or at least we thought we recognized, that these were the core components that we were going to teach ourselves and redefine the problem with the goal of how do we get to a clinical trial. A phase 1 clinical trial is the bottleneck that we have to get the science and technology ready to do before we bring it to a patient. That's why it's preclinical. Preclinical is 4-legged patients. 4-legged patients usually have whiskers and curly tails. Some of us call them pigs. Others of us call it preclinical. So this is a key component to this research process to be able to get to a clinical trial and then ultimately go through a systematic stage of product development that we actually create a new product. So success isn't one of these projects being a paper or a grant. Success is that paper and grant leads us and accelerates us all the way to that. And that's the grandiose vision that you don't do with an NIH grant, you don't do with an institution, you do it with philanthropy and partnerships. So where are we at in this model? We are actually right here entering phase 2 of our first product of cord blood stem cells. And that has happened in 8 years. So 8 years, we've gotten to that point with this focused mission. I think we're beginning to get confident that we can do this if we have the right people, the right team, and the right funding. Let me tell you a little bit more. I'm going to come back to the genetics at the end because I think this is an area where universities have a huge opportunity to deep dive in and make a meaningful contribution to this, and I'll give you an example of that. But I'm going to talk about stem cells for a bit here because that's what I get excited about. Anybody hear the recent papers out of Harvard? Anybody know this story? I don't have to spend too much time on this if you haven't heard of it, but there was one of the founding members of cardiac regenerative medicine, if you will, cardiac stem cells, related to what I'm going to talk about today, that worked at Harvard and recently had 31 papers retracted for being falsified. 31 papers. It sets the whole field back because now nobody trusts people like me because we all are crooks and we all are making data up and we're falsifying it. I think it's really awesome that Harvard University was bold and honest and retracted the 31 papers, but there is a wave of retractions now that are happening because of a few bad individuals making bad decisions. So science is a debatable sport. There's nothing black and white about this. It's shades of gray, but when we do make mistakes, we at least have to admit them and have to fix them. And this story will be told for generations of how individuals falsified data and tried to propagate, to publish, and pushed it to the point where it's completely falsified. And that's been done in many areas of science, and that's what we have to collectively buffer ourselves against. I can offline share some stories that I was in meetings in 2002 where that whole story could have been averted if people made the right decision. So it's a reality. It happened about 2 weeks ago. All of the research I'm talking about today is not related to that and is different, and I'll share with you how that is. So this is the vision. How do we take stem cells? How do we plant the seeds? And how do we grow new heart muscle? Anybody ever done this experiment with amphibious where you cut off a leg and watch it regrow? Anybody ever done that? No? Seriously? I did that in high school biology. Nobody's ever done that? So amphibians, they have this remarkable ability. You can literally cut their leg off and it will literally regrow everything, okay? How many times can you cut that arm off and it will regrow? It's not known. Every time it's been cut off, it regrows. So there is progenitor cells at the inflammatory environment at this cut surface that can literally regrow all of the different tissues of a limb, which is quite remarkable. Well, it's also true in mammalian species where if you cut the heart of a newborn mouse off in the first 7 days of life, it will grow back and have normal tissue, normal pink tissue. It will regenerate if you cut the heart of a mouse in the first 7 days. But if you do it after 7 days of life, you get this blue fibrotic scar tissue. So it loses its ability to regenerate and it gains the ability to become fibrotic within about a week of life of a mouse. So at some point, mammalian species can do this, but at some point we generally think we lose it. Y'all can cut your skin and you'll regenerate it. You can have open heart surgery in an 80-year-old and it will regenerate. But what do we know about the heart? Now we know the heart can actually regenerate in all of us. And this is what I'm going to tell you in the next minute or two of why this is important. So who was born in the 70s? Please, somebody raise their hand with me. Thank you. Just 2 of us in the 70s here? Really? Well, at least 2 of us are part of the greatest generation in the history of the planet for regenerative medicine. Okay, and let me tell you why. Because in the bomb testing area of the 60s, late early 70s, this world has seen the highest levels of carbon-14 that's ever been recorded. So carbon-14 in the atmosphere was increased because of the above-ground nuclear testing. And you can see as you plot this with then the predictable exponential decay of carbon-14, and you can do what we do with dinosaur dating. You just measure carbon-14 in any organic compound, and you can precisely date the birth date based on the carbon-14. In other words, if a cell was born in the 70s, this is going to be its level of carbon-14. If a cell was born in the 2000s, this is going to be the level of carbon-14 in that carbon structure of the organic material. Okay? Makes sense? So now you can take any carbon material, grind it up, get a carbon-14, and you can calculate its birth date. Fair? So with this idea, let me jump to the punchline. We're going to grind your whole body up and do a carbon-14 dating. What is the age of your body? Now you're thinking, well, is this a trick question? I know what birth date was.
Right?
What's that?
It regenerates. So when you have does cell regenerate at different levels of frequency throughout the body? So does it average?
Exactly. See, you're picking up on the thought exercise here. So the body does regenerate. Your red blood cells are 3 months old, so they regenerate every 3 months. So your red blood cells were formed with a carbon structure that's down here, right? What's the oldest cell in your body that never regenerated? Brain's a pretty good example. There's better examples. Anybody else? Oldest cell in your body. I'll even help you. I only want the females to answer this question. What's the oldest cell in your body? The oocytes. Did you know this? That all oocytes that your body will ever produce, you were born with? Right? Guys in the room, how old are your gametes? It's just biology, guys. It's just biology. They're months old, weeks to months old. So I always like to say this. You see where the joke goes. That's why we have immature guys and mature women, right? It's just biology. We all have our excuses. So the cells in the female gametes are as old as the chronological age. The gametes in the male body are very young because they're regenerating and producing new. This is biology. So take all of that together. What's the answer to the question? How old is your body all averaged together? Throw a number out there. And let's assume we're all 75 years of age right now. How old is your body when you're 75 years of age? 29. I love it. Bold answer. What do other people think? 10 years. But all averaged together. I think they call this calculus with an integral, don't they? I forget the area under the curve stuff. I don't care about your answer. I care about you thinking about it, right? So you're picking up on the thought process. Here's the actual tissue level. If you go from the youngest, the small intestine, the epithelium is 2 to 4 days old in your body right now. Epithelial cells are 2 to 4 days old in your body, right? The oldest cells down here, the oocytes, the female gametes, lifetime. The lens cells are a lifetime. And so you can see the estimated, where's the heart muscle in here? Right here. They regenerate about 0.5 to 10% per year. So there is a turnover of your heart muscle. I'm not going to go into the biology of that, but this gives you this idea now that I was not taught when I was at Eau Claire because it wasn't known, that the body regenerates and different tissues at different level. Your body is estimated to be about 7 years old. Think about that. No matter your chronological age, your body is regenerating. And the cells in your body with a carbon-14 dating are estimated to be about 7 years old. Does that not give you some spring in your step and give you some optimism? Right? That's a cool thing. The young people in the room are thinking, yeah, whatever. There's some of us in the room that thinks that's a pretty cool thing. So the human body does regenerate. That's the lesson out of this. And how do we start? I'm going to skip through these. Sorry, I've got too many slides here. How do we think about the human body? Well, here's my dramatic photo video of the day, guys. What you're looking at right here is, I think, the coolest thing I'll ever do in my career. This is what? Tell me. What do you see? A beating heart. I didn't tell you anything. I just asked you what you see, and you described a beating heart. What you're actually looking at is a microscope right now, and you're looking at about the tip of a ballpoint pen under a microscope, 4x power, okay? And you are seeing stuff beat and contract, and it actually looks like a heart even. It's kind of creepy, right? But this was 4 months earlier a piece of skin. This was literally a piece of skin. This was literally the skin from Bill Weir, the ABC Nightline reporter that came and visited us at Mayo Clinic, where we took a biopsy of his skin and we did this reprogramming technique and we coached it to be a stem cell and we differentiated it into heart muscle, and that's what you got. Now you're saying, is that really heart muscle? Well, I'll tell you this, if we put adrenaline on there, flight or flight, that will speed up. If we put a beta blocker on that, we'll slow it down. If we do a calcium measure, if we do a laction potential, this is cardiomyocyte. This, in fact, is Bill Weir's cardiomyocytes. It's genetically identical to him. This is proof that we can train our bodies to regenerate the heart. We can train our bodies to regenerate probably any tissue in the body with the bioengineering technology that's available to us. So our body doesn't normally do it because we might form cancer if we do it too much, but our body is able to regenerate. And if we could learn those tricks, could we actually take a congenital heart baby's cells, make their heart muscle, and transplant it? Well, that's the vision, a simple nutshell vision. Through a regenerative medicine, if we can understand those pathways that lead to regeneration, can we augment that? Can we strengthen that? Not going to go into the details, but I'll show you what we're doing. To do that, we are actually starting with babies with congenital heart disease, and we're collecting umbilical cord blood at the time of their birth. This is a photo of their first open heart surgery when they're doing it at 3 days of life. In that process, we freeze their cells, we store it, we do all the release testing, make sure there's not a contamination, that the cells are what we think they are. And at the 3-month surgery, we simply use a needle delivery device where we inject the cells back into the heart. Now, unlike the papers that were retracted recently that I alluded to, in 2001, people argued these cells that are being injected actually create new heart muscle, that the stem cells that we're collecting, processing, and injecting are creating the new heart muscle. We now know that's untrue. I could have told you that I believed it was untrue in 2003 and 4. The field debated this for the last decade. Now that has been falsified. What most of us field believes today is that the cells, the stem cells we inject into this heart muscle are actually more like fertilizer. They train the heart muscle itself to regenerate that it normally does at 0.5% every year, and it stimulates it to be maybe 10%. And so these stem cells that go in don't create heart muscle themselves, they create the ability of the heart to regenerate. And that's what cord blood stem cells and bone marrow stem cells do. It's fertilizer. But with that fertilizer, we can also collect it out of the bone marrow as shown here. We can also go in a different approach and we can put it into the coronary, the blood supply of the heart, and think of these green fertilizer packets are actually stimulating the underlying cardiomyocytes to divide and to grow. That's what stem cell therapy has been for the last decade, is not creating muscle itself, but is fertilizing the heart to differentiate on its own. That is different than the video I just showed you 3 slides ago, where we actually create beating, contracting tissue from a skin bioengineered sample. We can actually do that now as well. That has never been tested in a patient. That is what we are working on with the FDA right now, to be the first in the world that actually takes beating, contracting, bioengineered cells and put them into this deficient heart. So that would be like planting a seed of the cardiomyocyte and growing new heart muscle, where this is more like planting the fertilizer to stimulate the heart. And probably both together could ultimately help us rebuild the heart. So we do this with partners. In this case, I'm showing this slide because Regen is a company that was spun out of Mayo Clinic that actually does the stem cell manufacturing for us. We can't do that in academia. We can't do that efficiently. We have a partner that we work with that does that. And out of this effort, we've now treated 37 patients. The 38th patient is going to be treated on this coming week, next week. So this is the activity that's going on, that this is not theoretical. This isn't in 4-legged patients. This is actually in 3-month-old babies that this technology has evolved to, and we've treated dozens of patients with these types of partnerships. We can't do it without these 2 individuals, which is Todd and Karen Wanek and their family that has invested transformative amounts of dollars to allow our team to do this. But we also can't do it with just Mayo Clinic alone. We have a partnership with Philadelphia, with Children's Hospital in Colorado. This is Children's Hospital in Minneapolis and Children's Hospital in LA, as a consortium of hospitals that are working together that give us access to more patients, that allows us to do these things. So this partnership, I think, is really important to allow us to do things differently. Corporate partners and other institutions together is what allows us to take ideas and go through the animal work that I didn't share with you today and get all the way to clinical trials. It is possible if you have the right funding source and if you have the right partnerships. So we're the world's largest dedicated research team in 7 years focused on HLHS. We have the first FDA clinical trials in this area that we did in 2.5 years. We have over 1,000 active research participants in all of the research studies that are ongoing. And this is an old slide because last time I presented it was 28. Now it's up to 37. So that's a nutshell of what we do and how we have multiple partners working together. What I want to give you one other slide with to tease you what we could do at University of Wisconsin-Eau Claire in this types of ecosystems is focus more on congenital heart disease. So let me bring up one other slide and see if I can pull this off. There you go. Look at that. I love Apple TV. You guys are awesome around here. So here's a geeky slide. Let me tell you where this slide came from. What you are looking at here is our current view of what causes hypoplastic left heart. The molecular mechanisms of what causes in the embryo to form congenital heart disease of hypoplastic left heart. And let me tell you how we got here last week. And it's been an 8-year journey that I'm going to summarize in a minute. We've taken all of these 1,000 patients and we've done whole genome sequencing. Anybody know what whole genome sequencing is? It's literally the 3 billion base pairs of DNA in every single cell. We've sequenced every base pair in all of these patients that we have access to, moms, dads, and probands. So we have all of this genetic information. Did you know that your body is predicted to have 10, maybe to 100 lethal mutations? Lethal mutations in your body? I always like to tell people, you're the best looking dead people I've ever seen, right? You're predicted to be dead with the mutations that we all have. We don't know what all this information is, but bioinformatically, we have mutations that are thought to be not so good. So what do you do with that? Well, then we take these iPS cells, the stuff that skin cells, the beating cardiac cells I showed you, we make your heart, we look at the gene expression out of the heart, and we can take these thousands of variants that your body has, and we can start looking at which genes are differentially expressed in your cells in the lab. Make sense? Genes, now the transcriptome, the expression of the protein in your heart muscle, we can say, are you similar to your mom and dad or to your siblings? So that allows us to refine the hypothesis, but we still have hundreds of candidate genes that are mutated in your body and they're differentially expressed in your heart compared to your mom and dad. What do we do with that? You know what that picture is right up there? I love telling this story. God bless him. Dr. Lim. There's probably nobody in this room that remembers Dr. Lim. Do you remember Dr. Lim? Dr. Lim was a fruit fly geneticist here. He was one of the founding members of the hop gene, the hopping, jumping gene idea. Did it all in fruit flies right over in that grand building we call Phillips Hall across the street here, right? What we have done now with these hundreds of genes that have differentially expressed in the beating cardiac muscle of kiddos is we've gone into that fruit fly and we've knocked them out in a fruit fly. To date, we've knocked out about 600 genes in a fruit fly. That's a crazy number, guys, 600, that are predicted to be causing HLHS, that are differentially expressed and now knocked out in a fly, okay? And we do echoes on a fly. Echocardiogram, just like we do in the clinic, we do an echo on a fly, and we can actually measure the heart function of a fly. Is it normal or abnormal? And when this candidate genes from our patient population are knocked out in a fly, now we have an enrichment of pathways that we now believe are actually functionally causing a defective heart in this model system. So that refines our candidate genes down to what you see right here. What you see right here are the blue genes, and specifically ApoB and LRP2 are 2 genes that are in lipid metabolism pathways, never associated with congenital heart disease. It's mutated in our patients, differentially expressed in the bioengineered cells, and when knocked out in a fly, create a lethal cardiomyopathy. And it's thought now with the pathways that we've mapped out to be a p53-dependent cell proliferation pathway that this new biology is teaching us. Why do I share this slide with you? Because I think there's groups that do fruit flies, zebrafish, and other model systems where you take these candidate genes that come out of our clinical cohorts and we can start testing candidate genes in very functional assays that a lot of academic labs have the ability to do, but they don't know where to focus. What we can do with the Mayo's infrastructure is give candidate genes that we believe are high quality candidate genes driving human disease and test them in very specific model systems that university faculty and students are very good at. And this is the type of thing that we think will continue to evolve and give us a common final pathway and ultimately give us a mechanism of what all HLHS patients are dealing with. So this is really exciting because this is really mechanistic, and it's really the fruit fly team in Sanford Burnham that really uncovered this with us in partnership. So I'm not going to show you more slides than that because I could go on and we don't have enough time for that. But this hopefully gives you a sense of what we do from a research perspective and how we have a vision of going from basic research to clinical practice with research and innovation working together, doing it in the SpaceX way with the new generation, finding collaborations from corporate partnerships to university partnerships and hospital partnerships, and drilling down into very mechanistic studies like this that now you can start imagining targeting drug therapies and other therapies to address these molecular defects. So this is what I believe we can start doing together more and more. And I'm happy to take questions and learn from all of you of what we should be focusing on. Karen, you get the first question.
So I just want to make sure I understood. So the mechanisms that you're bringing now to clinical trials are they're distinct from this, in that this is going to focus on the cause of the disease, whereas what you're doing now is just trying to do repairs.
Yeah. It needs to be connected, and so I didn't take the time to connect these dots sufficiently, so I appreciate your question. We talk a lot about cause to cure, cause to the cure. And what I would be able to tell you if I had another half hour is that this cell proliferation idea really excites us for the following reason. Because when we put the cord blood cells into a pig model of hypoplastic left heart and we test the regenerative potential of that fertilizer, what we've been able to show in double-blinded, randomized controlled fashion, which is really important, double-blinded, I don't have to trust anybody that they're telling me the right thing because it's blinded. When you do it the right way, we've shown that the hearts of pigs have a 3-fold increase in cardiac proliferation when this fertilizer packet goes in the right window of time. Okay, so we can actually show that the cord blood fertilizer is able to stimulate a regenerative response. This genetic pathway, zooming in on the same pathway, now allows us to refine that hypothesis, and maybe you could even say we don't need cord blood in the future. We need a sonic hedgehog agonist and a Wnt inhibition in combination at the right time to get the same effect. So that's what's exciting, is when you start getting the cross-pollination of different model systems, that we do believe the heart regenerates, we do believe we can stimulate it, and now it allows us to refine that mechanistic understanding based on the genetics.
I missed a step, so could you go back and just clarify if there's that window of opportunity in the first week for the heart to regenerate?
Yeah.
But it's not until 3 months where the cells are
great
going
question.
back in. So I guess if you could just clarify what I missed there, and then the second part of that is, how are these kids doing? What preliminary results are you seeing?
Yeah, so you didn't miss anything. You picked up on exactly what I told you, but I didn't tell you the whole story. So
Okay.
I told you in the mouse it has a week to do, so we tested this in a pig because we don't know how long the human heart can regenerate. Nobody's done the biopsies to answer that. But recognizing that the pig is a good physiological model for humans, we've actually done a very careful BRDU labeling of DNA synthesis day by day with pig development. And what that data, it's not published yet, shows us is we actually have about an 84-day window of time that at 84 days the pig heart regenerates and grows just as fast as it did at day 1. This is exciting, right? Because these kids are having these open heart surgeries at these time points, and we're taking that regenerative capacity for granted. Is there something we can do to protect and preserve that regenerative capacity that we're damaging today? Is a great therapeutic strategy going forward to make things better. And that's probably leading into your second question. How are these kids doing today? I can tell you in the first 10 kids, 3-month-old babies that have received stem cells compared to counterparts that have not received stem cells, all 10 kids have gone out of the hospital with normal cardiac function. All of them have gone out of the ejection fraction is normal. 2 of them came into the hospital with abnormal ejection fractions. And all of the kids that did not get stem cells went home with an abnormal cardiac function. So there's something very acute within months that is happening. It's probably not the regenerative capacity. There's probably also anti-inflammatory mechanisms at play. And so now we're going back to the animals and understanding what else could be explaining this. But we've had a remarkable improvement that 10 patients, we have to scale that to 100 patients now to be more confident in, but it gives us wind in the sail to keep doing it. But most importantly, we haven't done any harm with these cells. And so we think it's a low cost, cheap, easy, feasible thing to do. And so now we're scaling it in phase 2 studies to try to prove that efficiency.
Do you need to bring it up earlier? I mean, instead of 90 days?
Yeah.
Think about doing it at 60, since you don't seem to know what.
It's a great question. So why did we pick to put the cells in at 3 months rather than 3 days? The answer was the following. We wanted to be 100% confident the product wasn't contaminated. It was what we thought it was, and so we had time to do the testing and the sterility We didn't want to put a contaminated product into the baby's heart and cause a problem. So we actually picked 3 months as a window that gives us time to test the product and show that you can inject it into the heart without causing harm. I can tell you the first time we did this, I was there pushing the syringe when we did it the first time. We do a lot of crazy stuff in healthcare, right? Cut people open, do all kinds of repairs. There wasn't a single person in the room that was breathing when we injected these cells the first time, because you don't know what's going to happen. We did it 40 times in a pig, but you don't know what's going to happen the first time you do it in a patient. Now we do it, we take it for granted. It's carefree, we're injecting cells, and we have the confidence we're not going to do any harm, right? But that first few times you do it, it's a big step, it's a big leap. And so you have to have all the preclinical data to have the confidence to do that, but you still don't know when you do it the first time. So, are we going to move it back to the Norwood? That's what we hope to do. Earlier the better, but we had to pick a time where we had the most confidence in our product because we didn't know what was going to happen the first time we did it. Great questions. You're picking up on all the gaps I gave you. Damn. Advice, thoughts. How do we do this? How do we do this university partnership with Mayo, and what are the opportunities for students and faculties, and what should we dream about in the spirit of Space X? Is hopefully the message you walk out of here thinking about. I spend my Wednesdays here on campus in Centennial Hall, 3rd floor, and so I always welcome and encourage people to come see me and visit me. We can put you on the schedule. But every Wednesday I try to make the afternoons here, and the mornings I'm over at the hospital. And I can do that because there's a shuttle bus from Rochester, Minnesota, where I live. And I come on Wednesdays and go back on Wednesdays. So I'm here, and I'm happy to be here. So thanks for being here today. Thanks for your time.